All right, thank you all so much for joining us today. I am very pleased to have our colleague, Justin Bopp, who joins us today from Stony Brook, where he's in the Department of Marine and Atmospheric Sciences, and you are a PhD candidate, correct? Mm-hmm. Yes. And I also can tell you, well, we met Justin, actually this past summer. I know a little bit about Justin. When we went to China for a trip to study horseshoe crabs with some of our colleagues, and I can tell you that he does a really haunting rendition of September and is completely shameless at it. So if you want to spend some time with Justin, you might be able to convince him to do that. But in lieu of singing, I don't know if you plan on any singing today.
Oh no, not today. I'm going to spare everybody their eardrums for that.
But he's going to tell us a little bit about trophic population and movement ecology of horseshoe crabs.
Yeah. So, well, thank you, Ruth, for the introduction, and thank you all for coming to my talk today. I really appreciate the hospitality that you guys have given me in this trip. It's been a real pleasure. And today I'm going to be talking about mainly habitat connectivity and different survival rates across many spatial temporal scales. And the reason why I have blue bloods in my title is because for those of you that don't know, horseshoe crabs have blue blood, and they're the true royals of the sea. And I'll tell a little bit about their importance there. But I just want to provide you a perspective of my outline through my talk today. So the first part of my talk is going to be introducing you to some animal movement theory and understanding the relationships and mechanisms that are at play when it comes to driving animal movement patterns. And then I'm also going to be going into a little bit of a background regarding horseshoe crab biology and management and their importance from both human perspectives and ecological purposes. And then the meat of my talk is going to be looking at differences in movement and survival parameters across different spatial scales, starting along the entire U.S. East Coast, then at the New York scale, and then finally at the bay-wide scale within a bay in the New York region, seeking to examine habitat use across juvenile and adult age classes. And then if I have time, I'll go into some future work that I plan on doing as well. So over the past few years in the field of movement ecology, there has been a lack of understanding uniform factors and mechanisms that underpin animal movement structure across many different taxa and organisms. And I wanted to present this conceptual framework that was initially established by Rand Nathan and colleagues in 2008, showing you potential interactions and mechanisms that can drive movement paths across these different spatial temporal scales. For those of you that are looking at this upper left-hand corner for external factors, we all think of this as environmental factors that can drive animal movement structure, such as ocean currents, temperature, salinity, and it can ultimately drive distributions of different animals across many scales. And then environmental factors can interplay with factors such as internal state, which could be animals gaining energy, as well as maybe circadian rhythms or circadian rhythms in their internal physiological structure. And then we also have navigation capacity, which is influenced by both internal state and environmental factors, which essentially ask the question, where do animals move and why? And then there's also another factor known as motion capacity, which essentially is the mode of movement that an animal may exhibit, such as swimming, flying, etc. But today, I'm going to only be focusing on one movement mode, which is swimming for crabs. And then ultimately, the combination of all these factors can drive the movement path of an animal. So this is just a really nice framework that I'm going to be emphasizing today across these different scales in terms of what we can elucidate in terms of understanding animal movement. And it's also important to not only assess mechanisms that drive movement, but to understand behaviors that may be occurring across these different spatial temporal scales. So again, from Rand Nathan's paper, there's a nice conceptual framework showing you what factors and behaviors you can elucidate across these different scales. The first one being is that at a fine spatial temporal scale, maybe over a course of a few minutes or hours, you may be able to elucidate behaviors such as resting or direction of movement. And then as you head into an increasing spatial temporal scale, you can further elucidate variation in behavioral switching, such as escaping a predator, understanding spawning, as well as searching for food, and etc. And then as you get to the overall lifetime track from birth until death of an individual, you can understand and interpret different behavioral modes throughout its life and ultimately understand population structure and mechanisms that drive that. And I just have this quote here by Rand Nathan, which describes like a nice overall synthesis of why understanding movement is important. And his quote states that movement of an organism is defined as a change in the spatial location of the whole individual in time and is a fundamental characteristic of life, driven by processes that act across multiple spatial and temporal scales. So ultimately, it's important to understand factors that influence movement across these scales. And it's also important from another ecological and management perspective to understand migration sources and sinks within several populations or regions. So here, since I'm going to be focusing on horseshoe crabs, I have a representation of a source and sink region. A source population in terms of a migration context can be defined as more emigration that's occurring compared to immigration, compared to a sink, as shown on the right-hand side in region B. It's important to understand these source-sink dynamics to help describe metapopulation dynamics, as well as define population boundaries for our managers to determine whether or not effectiveness of protected areas is working or not, and help also just decide if jurisdictional boundaries are actually relevant to the ecology of an organism. And you can also, by understanding these patterns, you can also begin to ask questions regarding mechanisms that drive these source-sink dynamics and migration patterns. And now that I've given you a full breadth of different reasons why understanding movement is important, I wanted to provide you a brief background regarding the importance of horseshoe crabs from human and ecological perspectives. So this is just a picture here on the left-hand side showing you that horseshoe crab blood is being extracted in a biomedical facility. And the reason why biomedical companies are actually interested in horseshoe crab blood is that they have this unique compound known as limulus amebicate lysate that is very sensitive to minute amount of gram-negative bacteria. So it's useful to ensure that medical supplies are truly sterilized. And the FDA has actually approved the use of limulus amebicate lysate since the 1980s. So it's been around for a long time, and there is a considerable harvest along the U.S. East Coast for biomedicine. As you can see here in the popular mechanics title, typically more than 500,000 crabs are bled. However, there is a variation in terms of the direct mortality attributed to the blood harvest. It's anywhere from typically 5 to 30%. But in terms of management, the Atlantic States Marine Fisheries Commission assumes that it's around a 15% annual mortality rate from the blood harvest. And horseshoe crabs also fulfill many ecological roles in their local ecosystems, such as maintaining migratory shorebird populations in the mid-Atlantic, such as in Delaware Bay. And the upper left-hand side, this is just a picture of shorebird aggregations in a spawning beach of horseshoe crabs of May this past year. And as you can see, these densities of shorebirds can be very high. And why they're there is because they're feeding on horseshoe crab eggs to contain enough, or to derive enough lipid sources for reproduction and survival. And on the upper right-hand side, this is just a picture of how dense those eggs can get. All those green dots represent a horseshoe crab egg that have been exhumed out of the sediment. So there's a lot of horseshoe crab eggs that are typically available to shorebirds in the mid-Atlantic. And I have a picture of the red knot here, which is an important migratory shorebird that is currently endangered according to the IUCN. And they critically rely on horseshoe crab eggs to continue the migrations from the Antarctic to the Arctic in the spring. And then also, horseshoe crabs are also fed upon by other predators, such as sharks, sea turtles, and alligators, as pictured on the right-hand side here. And they're also important for the bait industry across the U.S. East Coast. So horseshoe crabs are harvested in the whelk and eel industries. Oh, oops. Wait, what happened? Okay, press the wrong button, sorry. Let me go back. Okay, so they're important for the bait industry for whelk and eel pot fisheries. So those two images here to show you whelk and eel traps up above that graph. And the reason why I want to emphasize harvest is that there appears to be heterogeneous harvest among horseshoe crab stocks. So this map here on the upper left-hand side shows you the different stock assessment regions that are analyzed for survival patterns over a 5-year period on average by the Atlantic States Marine Fisheries Commission. So you have the New York region highlighted, or New England region highlighted in red, New York in purple, Delaware Bay in blue, and the Southeast in orange. And there appears to be heterogeneity in harvest when you begin to look at the commercial landings for the bait industry from 1998 to 2016, and the harvest landings and millions of crabs on the y-axis. And as you can see here, Delaware Bay overall, highlighted in blue, tends to comprise the majority of the harvest along the coast, with New York typically following in second, New England in a close third, and then the Southeast comprises the lowest amount of bait harvest. And I want to emphasize here is that bait harvest comprises the most direct mortality compared to the biomedical industry. And in terms of also understanding survival, recent stock assessment trends have also noted heterogeneity in terms of abundance levels of horseshoe crabs. And overall, since 2009, the abundance levels of horseshoe crabs on the coast have actually declined since 2009. So this chart here is just showing you the status of the coastwide abundance levels of horseshoe crabs as well as their respective stock assessment regions. And as you can see, the coastwide level is neutral, so it's overall. And by incorporating the abundance levels on the coast, it's doing okay, they're stable. But then if you look in the southeast, there's actually increasing abundance trends. Delaware Bay and the northeast are neutral, and New York is actually in a poor stock assessment status. So their abundances have actually been declining over the past decade in New York. So that's one of the reasons why we emphasize understanding these survival migration patterns in the New York area. So I want to give you an outline of the different data sets that I'm working with and incorporating different scales. The first one is that I'm going to be quantifying annual survival migration rates along the U.S. East Coast among these stock assessment regions to facilitate management and also test current movement hypotheses. And this data set comprises of 10 plus years of market capture data that comprises over 340,000 individual crabs tagged from 1998 to 2018. So it's a very massive data set. And then I'm going to be going into quantifying movement and survival. The same metrics that I'll be doing in the large scale, I'll be applying in the New York region through market capture approaches. And this data set comprises over 25,000 crabs tagged from 2007 to 2016. And then finally, I'm going to be, this is my smallest spatiotemporal scale that I'm going to be examining movement patterns. Specifically, I'm going to be asking questions regarding habitat connectivity and seasonal migration patterns among adults and juveniles in local embayment in the New York area. And this data set comprises of 95 individuals tagged with acoustic transmitters and have been tracked for movement patterns since 2017. So the first part, understanding these dynamics across the largest spatial scale, have a few objectives behind answering these or estimating these parameters. The first is that we want to estimate survival to compare survival with recent stock assessment trends to help managers identify areas of concern and determine if survival levels are sustainable in some regions versus others. We also wanted to compare our recent tagging data and the survival rates with the assumed adult-wide survival of 66%. Which has been derived from a multitude of different studies. We wanted to determine, specifically within the Delaware Bay region, if survival was below this mark, because this survival estimate currently is being used to maintain horseshoe crab and quotas for sustainable efforts in the shorebird dynamics in Delaware Bay. So we wanted to see if there was a new and improved estimate of the survival rate in the Delaware Bay region. And we also wanted to determine the survival implications with movement and effectiveness of protected areas, because there's heterogeneity in protections and harvests along the coast. So New Jersey has actually had established or has a moratorium established on horseshoe crab harvest since 2006. And then since 2008 in Delaware Bay, it's been established that males can only be allowed for harvest compared to females. And there's also a Carl and Schuster Reserve or horseshoe crab reserve in the mouth of Delaware Bay. And other areas don't have as much of these stringent harvest regulations. So we wanted to compare survival as well amongst these different regions with different harvest regulations and quotas. So, and in terms of migration, there's actually some disparate migration regimes among the stocks that have been observed from previous data sets. And we wanted to see if these trends were consistent with recent tagging data. And in the mid-Atlantic, the horseshoe crab populations tend to exhibit partial migration patterns. That is, they exhibit seasonal migration between the coastal embayments and the shelf. And there's a smaller proportion of individuals that remain permanent residents year-round. However, in contrast, the New England stocks tend to exhibit more localized movement patterns and highly philopatric populations. So we wanted just to see if there was actually any movement occurring between New England and the other stocks, and determining if there was possible partial migration. So many of you may be asking, how do we even begin to ask these different questions? Well, there's this handy-dandy coastwide tagging program that's been established by the U.S. Fisheries and Wildlife Service since 1999. And it mainly started out in the mid-Atlantic around Delaware Bay, but most states have participated in this tagging effort since 2009. And on the lower right-hand side, this is a picture of a tagged crab with a button tag. So you can see it has a unique tag identification number associated with each individual. And if anyone sees a tagged crab, they can actually input its recapture information in the U.S. Fishes and Wildlife Database. And the goal of this program is that we wanted to provide information regarding movement, longevity, and survival of horseshoe crabs. And we wouldn't be able to release this many crabs without the help of citizen scientists, which are our crucial effort in maintaining this tagging program along the U.S. And this is just a picture of citizen scientists tagging a crab within New York waters. And they help local management agencies tag and get measurement information. So our methods for analyzing this market capture data consisted of analyzing data from 2009 to 2018, simply because not all of these stock assessment regions were consistent in tagging efforts across years. There was actually gaps in tags released in some areas. And the total amount of individuals released during this time frame was 225,000, with a unique recapture rate of around 3.7%. And in order to estimate these survival and migration parameters, we employed multi-regional market capture models to estimate these survival rates among these different regions and look at movement between them as well. And we had to actually remove North Carolina and Florida releases because there was very low numbers of releases and recaptures, and there was missing gaps in releases over time. And we actually had to compare multiple possible market capture models, as we had different hypotheses regarding parameter constraints, which we'll get into in the next slide. So this is just a conceptual framework showing you how the market capture model works and estimate these different parameters. So we can start off by, in our first time period, initially tagging an animal, and then there's two possible fates that animal can endure in the next time period. Either it can survive, which is denoted by S, or it can die or permanently emigrate from your study system. And then if the animal survives, the animal can either choose to remain within a tagging area, or it can move to another area, represented by psi. And then from there, the animal can be either recaptured, represented by P, or the animal can be alive but may not be seen in your study area. So that's represented by the possibility of 1 minus the probability of recapture. And the parameters estimated in the model are survival, recapture, and transition. And we were mainly focused on understanding survival and transition rates. And then we can apply multiple parameter constraints, such as time-varying parameters or constant. And you can also add covariates such as sex, size, and other covariates of interest, and understand differences among these population parameters. And these parameters are estimated via maximum likelihood estimates. And in order to determine the best fitting model, we used AIC, or Keki's information criterion, to determine the best fit. And from our candidate model, we found that the best fitting model held survival and transition constant with time-varying recapture probabilities. And we felt that it was needed to keep recapture probability time-varying, given that there's differences in tagging effort among regions in terms of both tagging numbers as well as presence of citizen scientists. And what we found was that there was actually very little difference in survival among these 4 stock assessment regions, given overlap in the 95% confidence intervals. So you can see here, each of the regions corresponds to its color on the scale, and then we have survival probability in the y-axis. And although there was no differences in survival among these regions, we did notice some trends. In the New York and Delaware Bay area, you had lower uncertainty in your parameter estimate, and they tended to have the higher mean survival rates at around 62 or 63% respectively. And New England had 55%, whereas the Southeast actually had a lower survival estimate, but it also had the highest uncertainty around that estimate. And what was also interesting is that, remember, the 66% survival rate is actually used in some Delaware Bay models for horseshoe crab sustainability in terms of quotas. And it actually falls below that assumed 66% survival rate by a few percent. So it's about 4% lower. So this may suggest that there may need to be updates in terms of some survival estimates in those models. So overall, our implications are that there was no substantial differences among regions in terms of survival. But what was interesting is that there was trends for higher survival rates in Delaware Bay and the New York area, which is surprising given that 50% of the coast-wide harvest occurs between these two areas on average. But what may be happening is that these stringent harvest regulations, coupled with high densities in this area, may be buffering the impact of these higher harvest rates. So in New York and the Delaware Bay region, their horseshoe crab populations typically have higher densities compared to the northerly and southerly extremes. And there's also the lowest survival estimate in the southeast, which is contrasting relative to the recent stock assessment status, which is good for the southeast. But there's large uncertainty here, so we think that it would be maybe beneficial to come up with a more precise estimate of survival by providing more tagging effort towards the southeast. And in terms of regional movement rates, we did find that there was some heterogeneity in terms of immigration and emigration patterns. So this, I want to orient you with this table here first. So the rows represent the region of movement from, and then the columns represent the regions of movement to. And then the cells represent either residency probabilities within each region in the diagonal, and as well as movement to and from each state in the off diagonals. And there's a few interesting patterns noted within these movement patterns. One is that based on the mean migration rate, there was a total net emigration in the southeast that was 4.56%, which was much higher compared to other areas. And the southeast appears to be a small source to the Delaware Bay region, as there's 3.36% of movement that occurs from the southeast to the Delaware Bay. In the opposite direction, there is about 33% less movement. And what's also interesting is that the New York region had the lowest emigration noted, albeit that it's very small. And what's interesting is that if you look at movement from New England and Delaware Bay into New York, so you can see here it accounts for about 3% of migration from those areas into New York, whereas in the opposite directions, from New York to New England, it's less than 0.2%, and Delaware Bay, it's less than 0.5%. So it appears that New York may actually be a sink population from the New England and Delaware Bay regions. So our conclusions here is that there is overall relatively low exchange between stocks. Most stocks actually tend to exhibit high residency periods, as the lowest residency percentage was 95%. And movement into New York is 5 times greater than emigration when you account for movement from outlying areas. And we noted that the highest total immigration was in the southeast compared to New York, which had the lowest immigration. And we also noted that there was some movement from New England into other areas. So this could potentially suggest that these populations may not be as isolated as previously thought, because the only way to migrate into these areas is through the shelf. And there was overall migration preference for movement into Delaware Bay and New York overall from the outlying regions, such as the southeast and New England. So now that concludes the analysis of my large-scale patterns in terms of understanding population dynamics. But now I want to shift gears into understanding these survival migration rates among or within the New York region, because this is a priority concern for the United States Marine Fisheries Commission, given that it's in its poorest state according to recent abundance trends. And these highlighted regions here represent the different geographic regions we'll be estimating survival migration in between. These regions are highlighted on the left-hand side. You have the North Shore highlighted in red, the South Shore, which extends from Montauk to the Rockaways, just outside of Jamaica Bay. And then we also have the Jamaica Bay region highlighted in yellow, which is over here. And one thing I want to emphasize is that although this area is highlighted into New Jersey, we only included release information from horseshoe crabs in the New York area, simply because we did not have access to the coastwide data set at this time. So what I'm really saying is that we're looking at mainly release information from Jamaica Bay as well as Staten Island, from our market capture data set. And I also want to mention, too, that we analyzed this market capture data from 2007 to 2016. And over 22,000 individuals were tagged through this time frame with 879 unique recaptures. And from this data set, we formulated multiple hypotheses regarding survival migration, which I'll elaborate in the next few slides. And I want to mention that these results were actually published in Estuaries and Coasts this past July. So our first hypothesis has to do with differential harvest rates among these 3 subregions in New York. We noted that over the course of our market capture data set, 64.3% of harvest on average occurred in the South Shore area, whereas in the North Shore, there was substantially less harvest that occurred at 27% for the bait industry. And then we also had less than 5% of harvest occurring in the Jamaica Bay region. So given this heterogeneity in harvest rates, we expected that the survival would actually be the greatest in Jamaica Bay, given that there's low harvest here. And there's also actually a National Park Service that encompasses most of Jamaica Bay and the south shore of Staten Island. So there's lower harvest there, and then and more stringent regulations. And then between the two harvested regions in the South Shore and North Shore, we expected there to be higher survival in the North Shore given lower harvest rates relative to the South Shore counterparts. And hypothesis 2 has to do with sex-specific differences in migration. So we hypothesized that females would have lower survival rates compared to males, simply because in the bait harvest industry, females are assumed to be preferentially harvested given their larger average body size compared to males. And just to confirm that we had size as a sexually dimorphic trait within our study system, we have a size frequency chart here showing you prosomal width, and females did in fact appear to be significantly larger compared to males in terms of body size. And then hypothesis 3 has to do with movement patterns. So previous studies have actually suggested that there's either discrete populations in the surrounding area, such as in Delaware Bay, or less than 3% movement from Connecticut to New York over a period of 15 years. So we wanted to determine if recent tagging data actually supported this isolated movement trend noted in outlying areas, and to determine if there was more interbreeding and migration occurring between these regions. So what we found in terms of survival is that the highest survival, counterintuitive to our results overall, was in the North Shore at around 66% survival on average. And it was actually much different compared to Jamaica Bay and the South Shore regions. And what was interesting is that, remember, the Jamaica Bay region is predominantly closed to harvest, and there was very minimal harvest that was occurring there. And yet it had the lowest mean survival rate compared to the South Shore, although there was a lot of uncertainty between these two areas, so they were pretty much similar. But I just want you to keep that in mind when I discuss some survival implications in the next few slides. And then in terms of sex-specific survival, our models tended to support differences in sex-specific survival among these regions, with females tending to have lower mean survival among all of these regions. However, the largest difference in survival was between females and males in Jamaica Bay, with females exhibiting 24% lower mean survival rates on average compared to males. And this was interesting because, remember, harvest is not a big influence here. So, and we expected there to be larger differences in the South Shore and North Shore given that harvest is predominantly occurring in these areas. However, this may not be the case given that, especially in the South Shore, male and female survival is very close to one another. And in terms of migration rates, we did notice that there was moderate interbreeding and exchange among these different subregions, with the exception of low immigration between Jamaica Bay and Long Island Sound directly. There was overall less than 2.5% movement on average total between these two areas. What we did notice, though, between the South Shore and North Shore is that there was roughly 2 times higher migration rates that occurred, and compared to the reverse movement from the North Shore into the South Shore. And we also found the most stark contrast in migration patterns was actually between Jamaica Bay and the South Shore, as there was 19% movement from Jamaica Bay into the South Shore area compared to the opposite direction of very low exchange. So what this is suggesting that the Jamaica Bay region could be a source for the South Shore population, which is interesting given that this could be implicated in harvest regulations and management protections, especially since Jamaica Bay is closed to harvest and the South Shore is open to harvest. So there could be horseshoe crabs experiencing more susceptibility to the bait harvest in New York. And in terms of sex-specific differences in migration, our model did support some evidence for that. However, if you look at the migration rates between most areas across males and females, they tended to exhibit similar migration rates, with the exception between the Jamaica Bay region and the South Shore, as there was 10% higher mean survival compared to, for females compared to males in that same direction route. So our main conclusions regarding survival is that, again, we had the highest average survival pattern noted in the North Shore, with relatively lower survival in the South Shore and Jamaica Bay. And there are a few potential speculations as to why this pattern was occurring. One is that we may think that between the South Shore and the North Shore, the difference in terms of survival rate may be implicated in terms of both harvest as well as spawning behavior for horseshoe crabs. Because harvest typically occurs 1 to 2 weeks earlier in the spring on the South Shore, given that peak spawning occurs earlier on in these South Shore embayments. So what might be happening is that as the South Shore populations are getting harvested earlier and higher, there may be some conflicting management decisions that could infer higher survival for the North Shore, because as the 50% of the annual quota is reached on the South Shore, the daily harvest limits among New York State are reduced by 70%. So daily harvest limits start off at 200 crabs per day within New York, and then once 50% of the annual quota is reached, it goes down to 30 crabs per day. So what may be happening is that once the quota is being reached in the South Shore, the North Shore populations, by the time they're harvested, given late peak spawning abundances, they may be able to escape these higher daily harvest limits. And in terms of low survival in Jamaica Bay, we currently can't explain this pattern with our given data set. So it's worth exploring in terms of understanding factors that could be at play in terms of inducing this lower survival rate. But it could be due to anthropogenic effects, as there's been high heavy metal concentrations, eutrophication events, as well as a lot of pollution that has occurred in Jamaica Bay relative to the South Shore regions. And overall, we noted that there was slight differences in survival between sex among most regions, with the exceptions of Jamaica Bay. And we don't think that this could be due to differences in harvest preference in terms of a sex, just because the regions that were highly harvested had similar sex-specific survival rates. And in terms of migration, in terms of answering our third hypothesis, there was moderate exchange and interbreeding between regions. And the largest emigration pattern was noted out of Jamaica Bay into the South Shore. And there's a couple speculative reasons that we came up with in terms of explaining this pattern. However, we don't think some of them are likely. The first one is that there may be density differences between the Jamaica Bay population as well as the South Shore. So we were thinking that in order to maybe alleviate high densities around spawning beaches and intraspecific competition, we thought that crabs would be moving more to the South Shore where there's more open space and lower spawning densities. However, when we looked at spawning indice data over our time period, the magnitude and trends of spawning were actually the same. And then we also thought that this could be due to habitat availability and habitat quality, as, again, Jamaica Bay is more polluted. There's also just less area for horseshoe crabs to spawn in this area compared to the South Shore. So it could be a function of spawning habitat, as well as just maybe more habitat and resources to extract from the South Shore region. But again, our data cannot answer that for sure. And overall, there was little exchange between Long Island Sound and other regions, and there were slight differences in transition rates across sex, but mainly, again, in the Jamaica Bay region where the starkest contrast occurred. And now, going on to the third part, I'm going to be going into examining habitat connectivity across juvenile adult age classes within a local embayment known as Murch's Bay in the South Shore region of New York. And the reason why we focus on understanding movement patterns in this bay is that it's one of the most highly harvested regions within New York State, as over 22% of harvest actually occurs within Murch's Bay on average. And there's also high spawning abundances of horseshoe crabs noted. And recent data from my colleague Matt Scafani suggested that this could be an important migratory stopover site for the endangered red knot in terms of their reliance on horseshoe crab eggs for feeding. So there's multiple management implications that could arise from this bay and understanding horseshoe crab movement patterns. And before I go into our objectives for movement, I wanted to provide you with a brief animation in terms of understanding horseshoe crab habitat shifts throughout ontogeny. So juveniles, once they hatch their eggs and settle after their larval stage, after around 30 days, they tend to remain within the intertidal areas near adult spawning beaches. But then as they grow to around 4+ years of age, they begin to migrate into the subtidal area. And then once they grow and become sexually mature and to adulthood, they then migrate between the intertidal and subtidal areas on a seasonal basis. And the reason why it's also important to understand juvenile habitat use in general is that juvenile habitat use remains relatively elusive in the literature. It's been difficult to capture these larger age classes and size classes of juveniles in general, so it's been difficult to track them. And just to emphasize this elusiveness, I wanted to present you with molting data as well as live observations of juvenile horseshoe crabs across many different size and age classes in a bay in New Hampshire. And this is from Helen Chang's work during her dissertation. And what she found was that through scuba surveys and various other sorts of tracking surveys, is that she found that the majority of individuals she could find in the subtidal habitats were around 45 to 90 millimeters in prosomal width, and there was very few individuals that were above those size classes, which is important because this is the gap between juveniles moving on into adulthood. We wanted to determine if there was any differences in juvenile space use with adults, and just elucidate if there's differences in connectivity and dispersal. And in terms of understanding adult movement patterns, is that they actually remain very elusive in New York waters as well. For one, we don't know if individuals exhibit partial migration, and if they do exhibit migration, we don't know at what extent the proportion of individuals that may migrate versus remain as a resident contingent. We also don't understand cues that may influence the migration timing of horseshoe crabs between the bay and the shelf. So we wanted to examine potential factors such as lunar cycles, which are known to have an impact on spawning behavior, as well as temperature, which has actually been suggested to influence horseshoe crab spawning and activity rates. And actually, when Watson study through accelerometer and tagging data in Great Bay, New Hampshire, found that horseshoe crabs typically become inactive at temperatures less than 10 degrees Celsius. So we wanted to determine if this temperature threshold of 10 degrees was actually inhibiting migration activities or not. And we also just wanted to understand general habitat use and connectivity patterns across different season and sex. So we did this by tracking movement patterns of adults with our acoustic telemetry array in Murch's Bay. Each of these blue circles represents a receiver. The receiver is pictured here. We use Vemco, VR2Ws, and VRTXs. And these receivers we either attach to Coast Guard buoys or screw anchor moorings, as well as concrete moorings in the inlet. And the reason why we placed receivers in this configuration is that we relied upon the Coast Guard buoys, so it's irregular, it's not gridded, just given the fact that the buoys are not in a gridded array. Then we also have gates separating the eastern and western and central sections of the bay to look at movement and get directionality in movement. And we also have receivers placed in the eastern and western ends, as well as the inlet to understand migration patterns. And these gold and white stars placed on the map represent adult and juvenile release tagging locations. And we tagged 70 adults in June of 2017 with the Vemco V13 tags, and we used waterproof cyanoacrylate glue and a Velcro and zip tie attachment to tag them on the dorsal side of the carapace of the crab. And we captured these individuals by hand during the spawning season. And then in contrast, we tagged juveniles in the fall of 2017 to account for molting that actually occurs typically within August and September. So in order to at least gain a year of tracking data, we wanted to wait till most of the juveniles ended up molting in the bay. And we wanted to ensure that we could tag individuals that have just freshly molted. And we used smaller V8 or V9 Vemco tags, and we captured these juveniles via a crab dredge in certain areas in the bay. And overall, we had quite a fruitful return in terms of our number of detections and individuals detected. We had over 16 juveniles detected throughout the 2017 and 2018 period, and we had over 40,000 total detections. Whereas adults, we had at least 64 adults detected throughout our time frame, with over 230,000 detections occurring across these 2 years. And we're still actually currently analyzing their movements into the season. But overall, we typically begin to deploy the array in the beginning of April until sometime in November. With the exception of 2017, we had a delay in our shipment of acoustic telemetry supplies, so we had to delay the start of that year. So what we found was that there was actually evidence for partial migration. We noted that 22 out of 64 adults that were detected in this time frame migrated intermittently from Murch's Bay offshore, intermittently from July to November. And we also found that there was moderate bay-wide fidelity between years, as there was roughly 56% of those migratory individuals returning back into the bay in the spring of 2018. And we noted that there was actually one crab that actually migrated from Murch's Bay into Great South Bay in June of 2018. We also noted that there was some evidence for, and there was some individuals that migrated as well in 2019 back into the bay. We also noted that there was juveniles that also migrated. Roughly 8% of the detected individuals migrated from the bay offshore in November, whereas there was 0 returns from what our data could provide back in 2018. So this is interesting to find juveniles migrating offshore in this region. And we think that given that they have larger chromosomal width sizes, that they're getting ready to become sexually mature and move offshore. And what we found was that there appeared to be an influence of timing of migration and or temperature on the timing of migration. So here I have cumulative distribution plots across different seasons and temperature on the x-axis. And these plots have actually told us a lot about their movement patterns in regards to temperature. I just want to get you oriented with understanding one of these lines. So I'm going to start off with the fall of 2017 pattern of emigration to the shelf. And what we found was that if you look at this cumulative distribution function in red, over 50% of our crabs migrated at temperatures around 18 degrees Celsius or warmer. And all of them actually migrated above that temperature degree threshold according to previous hypotheses where horseshoe crabs become inactive below this temperature. So at first, we thought that, okay, maybe this pattern of this threshold is actually is consistent and across different regions. However, in the spring of 2018, we noted that there was a few individuals that migrated below this temperature, suggesting that there is some activity below this temperature threshold. And what's interesting is that the majority of individuals migrated at temperatures below 15 degrees Celsius. As you can see, it's around 60% of the cumulative distribution. So, and in the spring 2019, there was a similar pattern where most horseshoe crabs migrated at cooler temperatures. And according to Kolmogorov-Smirnov tests, comparing the fall and spring seasons and their temperature distributions, we noted that there was significant difference in terms of both fall and spring temperatures of migration. So overall, horseshoe crabs migrate at colder temperatures in the spring compared to their fall migrations overall. And we also wanted to look at the effect of lunar cycle, as I mentioned, on their migration timing. So what we found was that based on Rao's test of uniformity, that there was a significant difference in terms of horseshoe crabs migrating across lunar cycles. If you look at this rose plot here, you can see migration was not homogeneous across these different lunar cycles. And what was interesting is that the majority of individuals migrated at new moons. So it's suggesting that there is some type of lunar effect that's encouraging these migration patterns. So it'd be interesting to see from future data to see if this pattern is consistent or not. So overall, the migration of horseshoe crabs depends on temperature. They're across season. So most individuals migrate at temperatures above 10 degrees Celsius, but some do not migrate below, or some do migrate below this temperature. We also tested whether or not a photoperiod was a driver of migration using KS tests as well, but there was no significant difference of photoperiod. So it did not appear to be a strong factor in terms of influencing migration frequency. And we also noted a preference for migrating at new moons. And then in order to understand connectivity and space use, we employed 2 different approaches. One of these was spatial network analysis approaches, which is good for quantifying habitat connectivity, and one can derive multiple connectivity metrics from animal movement data. So this is just a simple network just to get everyone oriented. You can imagine these lettered circles representing an acoustic receiver, and then you can see that the direction of the arrows represents the observed direction of movement between different receiver states within your network array. However, network analysis does not account for preserving the time sequence of movement within your analysis. And it also doesn't provide a probability of connectivity. So we employed empirically derived Markov chain models to estimate probability of transitioning as well as remaining resident across different seasons. And it also preserves the time sequence of movement. And I just want to emphasize the case of where this Markov model has actually been applied in terms of understanding shark movements. This is actually derived from Stayfest and colleagues in 2015, where they used this modeling technique to estimate residency across different receiver locations in a bay for sharks in Australia. So our first network analysis metric that we quantified was degree centrality, and this is measured by the number of unique habitat connections that crabs make between unique receiver states or receiver locations. So those receivers with higher numbers of unique connections tend to have higher degrees of centrality, and this degree centrality metric can be thought of as a proxy for a level of connectedness between other habitats in your network. And I want to get you oriented with the next network analysis slide. There's a lot going on here. So what we did is we compared this degree centrality network metric across adult males on the left-hand side represented in red and females in blue on the right. And we wanted to look at seasonal differences in connectivity patterns between and within these sex groups for adults within the spring 2018, summer 2018 in the second row, and the fall on the bottom here. And each of these circles represents an acoustic receiver location, and it's graduated by the degree centrality metric. So one thing that I want you to note is that if by looking at adult males and females between or within a season, their connectivity patterns do not appear to differ substantially. You can see here that adult males and females tend to both exhibit very dispersive patterns of connectedness between the bay. They have wider spatial extent, and they utilize more habitats and have stronger levels of connectivity between habitats. But then as you progress into the summer and fall, you notice this pattern that is fairly consistent between both males and females, where they start to restrict their habitat use in a certain area of the bay. And then as you head to the fall, they tend to not utilize the eastern and western extremes, and they tend to still have some moderate connectivity in the central habitats of the bay. So overall, the central habitats appear to be relatively consistent for maintaining connectivity for both males and females. And one thing I want to mention is that we think that the reason for high dispersiveness in the spring of 2018 is that this could be due to temperatures beginning to warm, starting to induce greater activity patterns in the crabs, as well as they may be searching for suitable spawning mates and spawning habitats, because this is typically when their peak spawning occurs. And in terms of our second network analysis metric, we employed eigenvector centrality, which not only accounts for the number of unique connections a habitat has to a different habitat within your network, but it also counts for the strength of that connection. And you can think of this as a proxy for preferred habitat use. And this is just a simple network model here showing you receivers with high eigenvector centrality either have high numbers of unique connections or are connected to receivers that themselves have a high number of unique connections. And what we did is we compared eigenvector centrality across both adult males and females, as well as juveniles in the spring of 2018. And what we found was that, again, the more preferred habitat use for both males and females tend to be within the center habitats of the bay, and they tended to exhibit very dispersive patterns and high connectedness among habitats. But then what was interesting is when you looked at the juvenile data, they only moved between two unique receiver pairs. And the most important preferred habitat use was actually in this north central habitat in Murch's Bay. So there appear to be contrasting differences in terms of migration patterns between juveniles and adults. And you can also see this trend in the residency probabilities derived from our Markov chain models. So you can see adults in the spring of 2018 tend to utilize more habitats similar to network analysis, but their highest chance of residency at any given habitat within the spring period is around 10%. But then in contrast, we look at juveniles, they only occupy 5 habitats for residency periods, and the majority of their probability allocated towards residency was in the north central habitat of the bay in this time period. And in fact, if you compare the cumulative probability of residency in juveniles compared to adults, they have a 40 times higher chance of remaining resident relative to their adult counterparts in this section of the bay. So overall, juveniles tend to exhibit just more restricted space use and tend to have disparate spatial overlap compared to adults. So overall, our conclusions are that adults experience the greatest habitat connectivity in the spring season, and then they start to narrow down their habitat or their migration connectivity towards the central sections in the bay as you head to the fall. And juveniles, again, just had more restricted space use, less connectivity overall between habitats within the bay. And now I'm going to be going on into some future work that we're planning on doing next year. So we plan on going into an even finer spatial temporal scale. We plan on actually employing an acoustic receiver array in a subsection of the bay where juveniles are consistently caught, represented by that green square. And we want to tag juveniles again in a similar manner that we did with our current data set. And the beauty of this array is that you can actually estimate position through triangulation techniques and acoustic telemetry, and you can elucidate different resident, like transient and resident behaviors. So what we want to estimate is one, fine scale home range across different seasons. And we also wanted to determine if diurnal cycles as well as tidal cycles influence changes in behavior, such as transitioning maybe from higher activity, maybe for feeding or just extensive search patterns compared to residency or just lethargic periods. And we also want to look at the influence of more environmental factors such as like tide and direction of movement as well. And so bringing this full circle, remember that conceptual framework about the different factors that one can derive across these different spatial-temporal scales. We have external, internal, represented by these columns, and we also have navigation, understanding direction, and population parameters that we could estimate. And I just want to emphasize across these different spatial-temporal scales is that we could not estimate everything that we wanted to in terms of understanding mechanisms that influence movement. As you can see in the upper two rows, looking at our regional data set and our coastwide data set, we couldn't determine which external and internal factors influence these migration rates. But we could get at directionality, such as immigration and emigration patterns, as well as estimating survival. But then as we headed into the smaller spatial-temporal scale and the bay-wide habitat, we could get at lunar effects as well as temperature and photoperiod impacts on migration frequency. We could test previous hypotheses regarding physiological thresholds for activity, and we could also determine seasonal space use as well as compare sex differences in habitat use and connectivity. And then we hope to gain in this fine scale array is understanding additional external and internal behaviors and looking at the influence of their movement and home range and understanding more fine scale connectivity for juveniles. And with that, that concludes my talk, and I'd like to thank all of my collaborators from Cornell Cooperative Extension, Stony Brook University, and the USGS, as well as the Department of Environmental Conservation. And I'd like to specifically thank the New York State Department of Environmental Conservation for funding this research through the Ocean and Great Lakes Award. And with that, I'll take any questions.